Hi, my name is Alexandra Machrone. I'm a PhD student at Binghamton University, and today I'm going to talk to you about starvation selection and how it reduces mating success in fruit flies. So in nature, there's a common theme that bigger is better. Larger individuals show to potential mates that they're more fit and have better access to nutrients, and therefore they're a better mate. But that isn't necessarily always the case. In fact, starvation-selected Drosophila, which are much larger than typical fruit flies, have a severely reduced mating success. So starvation-selected Drosophila are flies that were taken off of food when they pupated, put on a low-calorie diet of agar, and starved until only 15% were left surviving. Then the survivors were placed on food to mate and lay eggs, and the process is repeated over and over for 100 generations, until eventually you end up with a fly that's resistant to starvation. So these starvation-selected, or S flies, have quite a few adaptive traits which allow them to survive starvation. They are larger, they have more fat storage, they reduce their activity, increase their sleep, and even increase their larval time so they can grow more before they pupate. However, they also developed some maladaptive traits, and what I'm focusing on is the reduced fecundity, specifically the reduction in mating success. So far, we've been trying to answer two main questions. The first question we looked into is physical size what limits the mating success? We thought that it could possibly be a mechanical problem and the flies might just be too large to be able to successfully mate. So we developed a protocol to reduce the size of the S flies to the same size as our normal fed F flies. And we came up with 4 different treatments for each fly type. The first 3 treatments worked as controls and they didn't actually change the final size of the fly at all. For the test treatment, we scooped the larvae from their vials and placed them on a low-calorie diet of agar. And these flies were unable to get sufficient nutrients as larvae, and the adult flies ended up being around the same size as our control flies. We did the same thing with our control F flies, and we ended up with an even tinier fly in the scooped agar treatment. So we did this over and over again in 3 genetically different fly lines. And as you can see here, the control F flies do very well, and they're mostly successful within 10 minutes. Whereas when we reduced the size of the F flies and we made that really, really tiny fly, they did very poorly. In most cases, they were even worse than the S flies, which we were trying to rescue. We also found that only line C was able to partially be rescued by reducing the size of the flies. And we actually saw a similar trend in every group where S flies were consistently worse than their F fly counterparts. The only treatment that we saw an opposite effect on was our rescue flies in line C. So our rescue S flies were actually about 4 times more successful than our rescue F flies, and twice as successful as our control S flies. So next, we wanted to find out if their courtship behaviors accounted for their reduced mating success. And in Drosophila, courtship is a series of complex steps. Males will begin courtship by chasing the female. He'll then approach her, he'll lick her, he'll walk around her and sing, and eventually he'll mount her and attempt copulation. And if they're lucky, they will mate. We recorded videos of flies during courtship and ran them through 2 programs to analyze their behavior. So Fly Tracker can track simple features and behaviors like velocity, leg position, and wing position, whereas Java, which is what we transfer them to next, is a learning software. So you can train Java to recognize certain combinations of features from Fly Tracker. And you can teach Java to recognize complex behaviors like singing, chasing, or attempting copulation. The first behaviors that we looked at were chasing and singing. We wanted to see if the S flies were doing these behaviors as often as the control F flies. And we found that there was no significant difference in any group for either behavior. So that means the S flies were chasing and singing just as much as the F flies, which is surprising, because we already knew that their activity levels should be reduced overall. Next, we figured if they're courting properly, then maybe something's going wrong right before mating. So we decided to take a look at copulation attempts. A successful copulation attempt should look like this, and it ends with the flies mating. But we found that more times than not, copulation attempts are unsuccessful. The male will mount the female, he'll curl his abdomen like he's ready to mate, and then he'll simply let go. Some flies will actually go through the whole courtship process, but they never attempt to copulate. So here we looked at the percent of fly pairs that attempted to copulate at least once, regardless of whether or not they were successful in the end. And what's interesting is we saw that exact same trend here as we saw with the percent of successful fly pairs earlier. F flies were consistently more likely to attempt copulation than any of the similar S group flies. And with the exception of line C again. In line C, the percent of flies that attempted copulation was almost equal to our control F flies. In fact, it's higher than any of the other S fly groups. Sometimes the flies are successful on their first attempt, and sometimes it takes multiple attempts until they can successfully copulate. And we see the same trend here with the F flies being successful in their first attempt more than their similar S fly counter groups. And again, we saw something different with line C. So our rescue S flies were successful on their first copulation attempt more than any other group. They were more likely to be successful on their first attempt than even all of our control F flies. So all of these differences here that we're seeing with line C tell us that each line probably has a different reason or reasons behind their reduced mating success. Whereas with line C, it seems like a big part of it was actually just their physical size. Finally, we noticed that some flies would attempt copulation by holding on to the female for a long time, but they would have fewer attempts overall. Whereas some flies would quickly attempt copulation, but then would try again and again and again pretty frequently. So this led to an overall similar amount of time spent attempting copulation, but we wanted to see if one strategy was better than the other. So we looked at the percent of copulation attempt bouts that each pair had that were greater than 1 second long. And we found that no matter what group you're in, longer bouts were more common in successful flies. So even though they tried the same amount overall, the successful flies were more persistent and were less likely to let go of the female even when she moved away. Although we see a similar trend in what makes the fly successful here, the proportion of S flies that have long copulation attempt bouts is lower than the proportion of F flies that have long bouts. So we still need to figure out why that proportion is different. Our next steps for this project are going to be to go back and look at other courtship behaviors and break them down into bouts like we did with the copulation attempts. We looked at the overall courtship times and the total amount of time spent singing and chasing, but it's possible that the breakdown of each bout and the persistence is different. We also want to look at non-courtship behaviors to see if this lack of persistence spans to other behaviors. It's possible that it's causing them to end their bouts sooner. Alternately, we also want to look at their cuticular hydrocarbons because we know that S flies have a different ratio of contact sexual pheromones, so it's possible that when they're coming in contact with the other flies, it doesn't taste right and they decide to abort the copulation attempt. We also know that food availability during courtship increases success in normal Drosophila, so we want to test to see if food availability is going to have any effect on the success of S flies. And finally, we want to see if there's any genetic differences that might account for the reduction in mating success. So I'd like to thank my lab, my committee, and my contributors, and that's it.